the camera for now. Okay, so good evening and a very happy new year. Um, Today we'll be resuming our discussion with regards to what we had already started in the last class. So I'll go through a quick recap of the discussion we did in the last class because it was a new topic, I think, for some people, and because it is in continuity with what I want to discuss today. So, just to refresh your memory, we were discussing the procedure or the protocol which is to be followed if you want to perform analysis of next generation sequencing data. Uh, Keep in mind that the protocol that I'll be showing here is it is a generic procedure that you need to follow for any NGS technique that you want to analyze or any NGS data set that you wish to analyze. And depending upon what type of analysis you're doing, some tools are more optimized, more preferred. But overall, the workflow or the core steps that I'll be discussing, they remain pretty much the same. Just Okay, now first we had done a quick recap of what exactly NGS data or the procedure that you need to follow with NGS data. So you start off with hypothesis designing, of course, and then based on what is your experimental plan, you follow the same and you choose the most optimal technique under those conditions, and then the output that you get as your sequencing experiment are raw reads and the FASTQ files that you get out of those. And the first step that you need to perform with that FASTQ file is to perform pre-processing, data pre-processing, whereby you um, check the quality of that data. And the second step that is part of that pre-processing is what we'll be discussing today. That is the trimming step with regards to your NGS data. Um, Next, we basically moved on to some of the key terminologies. For example, what do we mean by data? What do we mean by, I mean, on by data as in what are the different terminologies which are used when you're working with NGS data? We talked about fragments, the read, the two mates that you will get out of a paired end sequencing experiment, the depth of your sequencing reaction, how you should choose that, whether it is single end or paired end, and what is more reliable. Of course, paired end is more reliable under those circumstances. Then the coverage of your sequencing reaction, a particular equation which fits into that particular protocol. Then we moved on to FASTA format and the use of FASTQ files in a step known as FASTQC, which is a quality control step before proceeding with extensive analysis of your data. FASTA format, which you might already be quite familiar with, is a text format where you either can get a nucleotide sequence or a protein sequence, as you can see here on the right. And basically, it consists of an arrangement. It's like a string, basically, of ATGC sequences, or if it is a protein sequence, then it includes the single letter code for all of the amino acids. And let me switch this on. It's been meaning to otherwise. Yeah. So either a sequence of nucleotides or a sequence of the amino acids which are composing the final peptide or the protein. And usually that is necessary for most of the tools that you will use, not just for NGS data analysis, but in general, whenever you are submitting a sequence for synthesis or you're performing Sanger sequencing reaction, or the most standard format for these sequences is the FASTA format. Then we moved on to the FASTQ format, which is for a sequencing read. And it follows this particular step whereby you have certain defined notations. For example, you have an @ sign followed by the name of the sequence, then the actual sequence of the read, and the length of that sequence is already defined here. Then you have a plus sign, which is a separator, and the fourth line, which denotes the quality of your sequence, which is represented in ASCII characters. If you remember, we had next discussed how these ASCII characters are actually decoded. And they can be converted into decimal and hexadecimal characters as well, which denotes the quality, which gives you a score, which denotes how good that read actually is, how good is the sequencing that has been performed for that particular read. And based on that, you can actually filter which sequences you want to use for further processing and which you want to filter out. Following that, we had also understood the concept of Phred score, which is the probability that for a particular region of your sequencing read, the base that has been called or the base that it has been assigned is actually correct and is not an abnormality or a probability which has just gotten read by the sequencing machine or the sequencer as a chance event. Then the calculation for Phred score was also shown. Like for example, it is a segment of a particular read here, and that has been converted. Each of those characters has been converted into the corresponding ASCII character, and the summation of all of them denotes the final Phred score. And then the accuracy or the um, how correct or how accurate that sequencing reaction has actually proceeded can be defined by how high the Phred quality score is. The higher the Phred score, which is most commonly you would prefer a Phred score of around 50, then in that case, a Phred quality score of 50 means that the probability of it being incorrect is extremely low, and thereby the sequencing reaction's accuracy is extremely high. So that is what we most commonly see here. And you can see that it denotes around 99.999% accuracy in calling the base at that particular position. So that is what we saw here. Then we understood a few other formats, like for example, SAM. We will be understanding the use of SAM and BAM in the next class in more detail. If you remember, I had mentioned that they are mainly useful when you are aligning sequences to the reference genome. So this process of alignment, the tools which are used in it, how these formats are actually useful, why it is important to convert from a SAM format to a BAM format, and then use that further. All of that we'll discuss in the next class when we discuss alignment, because it's a bigger topic. I'll be briefly introducing it today, where we'll just start off with this with the concept of reference genome, and some part of it I had already introduced in the previous class, and we can take it up further in the next class. Then we discuss the GFF or the GTF format, which gives you which is defined as the gene transfer format or the general feature format, which are annotation files, which basically gives you the entire information about that read. So what do we mean by the information, so-called information? It includes all of the data, like whether it is the original sequence, whether there are any changes from the original sequence, whether there are any polymorphisms, any mutations, like for example, SNPs or indels or structural variants. All of that annotation is carried along with the sequence in the GTF or GFF file. And this all we have discussed already. It majorly consists of these kind of information. And you, of course, have the gene ID value. And if it is a coding sequence, you'll have the transcript ID also for the same. That within the genome, this particular locus encodes for this particular transcript or this particular mRNA. And that will give you the that will give you that will give more annotation to the sequence which has been decoded. Then we moved forward to the bed file, which we have discussed later on, which we will discuss and have discussed previously as well, which gives you a format for representing the features of the sequence that you have already analyzed. And most importantly, you will learn more about this when we come to discussions about ChIP-seq, because that is where it is more relevant. And you can actually use that for IGB as well, which is the integrated genome browser, where you can see the representation of ChIP-seq peaks for analyzing how well your already performed analysis has worked. Then these are the additional information, which is also part of the GTF or GFF file, where you have the coordinate position, you have any conservation, any variation, transposons, origin, transcription factor binding sites, etc. And finally, we talked about VCF format, which is the variant call format, which contains information about any variants which you can find compared to the reference genome. Again, this is a step which you'll understand in more detail when we have already discussed alignment, because alignment is going to give you that clear distinction that this is the expected sequence and these are the XYZ deviations from that expected sequence. And based on that, you can filter out whether a particular region is carrying a variant, whether it is a mutation, whether that mutation is disease causing, or it is simply, like a polymerase error that has no consequence, whether it is a silent mutation. All of that information can be decoded, and we'll learn more about it. Then we had moved forward to the various sorts of outputs that are found as the result of FastQC analysis. So if you provide a FastQ file or your or the information which is contained within your read as the raw data file after sequencing, if you give that to FastQC tool, then what does it do? It performs an analysis of those sequencing reads, and it gives you an output as to how good that sequencing read actually is, and whether or not you should use it in further steps of the analysis, and whether the sequencing machine has worked well enough for that sequence to be reliable. Okay. And what are the different parameters that it provides? Base statistics, per base sequence quality, per tile sequence quality scores, sequence content, GC content, per base N content, sequence length distribution, sequence duplication levels, overrepresented sequences, and the adapter content. So first, I'll not go into as much detail as we went in the last class. If you have any doubts, you can let me know and I can discuss that particular topic in more detail. But basically, the first output, which is something we primarily check when we are performing FastQC analysis, the first output that you get here is the per base sequence quality. So what does it mean? It means that for each position within the read that you're taking into consideration, how high or how good is the quality score for that particular position, whether or not it is reliable or not reliable. So here you can see that this gives you box and whisker values, and the ideal sort of output looks something like this, where most of the sequencing read quality score lies within the green region, which is a good score, a Fred 33 and above score. And what you see here are quite poor sequencing reactions, especially because they are much longer reactions up to 250 base pairs, whereby the efficiency of your machine reduces, the probability that errors can get incorporated, that increases, and overall the sequencing reactions reliability decreases by a lot. So this is the first plot that you get out of your FastQC analysis. Second, we looked at per tile sequence quality, whereby it represents how for a particular Illumina sequencing reaction within a flow cell, how well the sequence for each tile within that flow cell has actually proceeded. If the flow cell itself was defective in some regions, if it had um, some fault, or the adapters which are ligated in that particular region were not working properly, where the PCR didn't occurred properly over those regions, if there wasn't enough template amplification, and number of possibilities exist. Basically, in that entire slide, if there is any region um, which has not properly undergone sequencing, then any result which you get from such a tile will not be reliable and should ideally be gotten rid of. And basically, it gives you an analysis of how well the overall sequencing reaction has proceeded. Then you have your per sequence quality score, whereby you would want that the quality of your entire sequence should be quite clear. Basically, for each read, what is the average quality, and you would want it to be obviously quite high. So this gives you the total number of reads versus the mean sequence quality score over each full read, and it allows you to see if some subset of your entire sequences has a universally poor quality. And what you would ideally want is for most of the bases to have very high quality and only a few to be of low quality, which is something you can see here, in contrast to the first, where you can see a lot of bases have lower Fred score and yeah, it's not very ideal. Then the next parameter you get is the per base sequence content. So you can see here that for DNA and for RNA libraries, you get different sorts of outputs. For a DNA library, you would want that these two lines, they sort of run in parallel. What do these lines represent? They represent the percentage of A, T, G, and C in different colors. And basically, it shows you how much of the entire composition of that entire read is actually contributed by A or T or G or C. And of course, A and T are expected to be equal, and G and C are expected to be equal, which is what you see here as well. So, yeah, that is pretty much it. You would expect there should not be any difference between different bases of the sequencing run. So they should run parallel to each other. But if there is a bias and if certain bases are overrepresented or excessively present in a particular sequencing reaction, that means the polymerase probably has not worked properly and has just randomly detected some incorrect bases. And note that for DNA-seq libraries, the proportion of each base remains relatively constant over the length of the read. But for RNA-seq, you would not see as uniform a distribution for the first 10 to 15 nucleotides, which is the expected sort of outcome for an RNA sequencing library. But overall, you can see that eventually the percentage of each of these bases becomes nearly equal, and you can see parallel lines which run along each other. Then you have the per sequence GC content, where you want the GC content to be distributed, to have a normal distribution over all sequencing reactions. And that is, of course, the ideal expectation. If it is shifted too much, then that is not very desirable. Then you have per base N content, where you want to figure out how much of that sequence has at, or how many times at a particular position in a particular sequence a base call has not actually been done, and instead the sequencing machine or that reaction has annotated it as N, which means that it could not identify what is the actual base there, and it randomly just called for an base N where it can be any nucleotide. So the example here, it shows you that the report here shows you that it was unable to call a base for 20% of the reads at position 29. That means there's some error there, because of which the sequencing reaction is not proceeding smoothly and there was some sort of a problem. So this needs to be looked at. And I think that was our last slide for, yeah, the last class. And then we looked at sequencing length distribution, whereby in terms of short read sequencing, fragments of uniform length should be generated. And after trimming, sometimes this can fluctuate. Again, we'll discuss that today because of either poor base calls or adapter contamination. And if you have performed long read sequencing, you would have a lot of variation in the sequence length. And sequence duplication, you can see there are two sources of duplicated reads. One, because of biased PCR enrichment, because of PCR reaction proceeding inefficiently or in a biased manner, or very overrepresented sequences, such as very abundant transcripts in an RNA-seq library. So that also you might get sometimes, and you can see there is a percentage of sequences which are showing high duplication. So this is actually expected for an RNA-seq library, because you will find certain sequences to be highly enriched and to be present to be represented multiple times. But for DNA sequence, you would not expect that to be the case, and you would expect it to be for expect there to be only one copy or a maximum of two copies if it is diploid, and not more than that. Sorry. So this is what we had discussed in the last class. If there's anything you want to discuss from there, you can let me know. And taking that forward, we will be moving on to the next part of the discussion. So what did you start off with? You had raw FASTQ files, which are reads basically. And what did you do with them? You performed initial sequencing quality check, which is where you analyze whether or not the sequencing reaction has been performed properly. And the reads that you have gotten, the raw data files that you have gotten, are they of good quality or not? That is the first step we have done. Now for the second step, what do? We do a trimming of the reads. And in the third step, we actually align the now trimmed reads back to the original reference genome. And we will just briefly just understand the definition of the reference genome today. And that will be the discussion for today's class. Again, a lot of information that I discuss as part of this entire module, where we will be talking about the different steps of sequencing analysis. A lot of it has been taken from this particular book, Next Generation Sequencing and Data Analysis. And you can refer to that book for your own reference. In addition, today, what I'll be showing with respect to Trimomatic particularly is a part of their own manual. It is a tool which has been um, created and has been commercialized by USA Dell Lab. And that is what we will be taking up today. So you can refer to their manual also, where you'll find certain things in more detail. Um, Especially if you're someone who is handling the data on your own and you want to use the tool, then in that case, how to actually structure your code, how to give the command, that information you can probably refer to the manual in those circumstances. But today we'll be discussing the basics of the tool. We'll be discussing the various parameters which are present there, and we'll understand how to actually use it. So First, first thing that you should definitely understand is that trimming or Trimomatic, the example that I'm giving here, there are other tools also. Trimomatic is what most commonly we use. Trimomatic is also performing a part of pre-processing itself. We have still not moved into actual data analysis. We are still making our data more, we are optimizing our data for the analysis steps at a much higher, to be performed at a much higher efficiency. Now, what is Trimomatic? It is a fast, multi-threaded command line tool that is used to trim and crop Illumina data, basically fast Q files, as well as another job that Trimomatic very extensively performs, and which is very, very important if you are proceeding with data analysis, is to remove adapters. Why are we so bothered about adapters? We'll come to it. These adapters, they pose a very important problem depending upon what is the type of library that you've prepared and what is the downstream application. Look at this particular schematic on the right. We'll be discussing this in more detail, and some terms you might not understand right now. But basically, after sequencing, if this is the read that you're getting, this is your unchecked sequence. This is your adapter. Sliding window is a strategy which Trimomatic uses. We'll come to it later, how it exactly works. What is it doing? It is scanning through the sequence basically, and it is checking which sequence is actually valid and which sequence is incorrect. What will it do? It will check for known defined adapter sequences and it will remove them. Think about the fact that no, just wonder why an adapter sequence needs to be removed. First of all, why was it attached? It was attached because we needed it for template amplification. You remember where we had a flow cell, and on top of that, you have these adapter ligated probes, which are just, sort of standing on top of the adhere to the flow cell. Now, to bind to that or to hybridize to that, your sequence also needs to have a region of complementarity. That complementarity is defined by these adapters. Go back to the Illumina video and that will help clear up this concept. Now, while it was necessary for template amplification, why is it necessary to remove that? Why are we calling it contamination? Because that sequence is not something which is inherent to the cell. It is not a part of the cell's DNA, not a part of the cell's RNA, or it's something you are not interested in. Secondly, especially if you are working with RNA sequencing data. In that case, if you retain RNA or if you retain adapter ligated RNA and specifically the adapter sequence within that RNA, what it is going to do is that for so many reads, you will have a lot of adapters which are actually in common. Under those circumstances, when you perform analysis along with your actual results, you will also end up detecting these adapter sequences, right? Because those sequences are, I mean, think about it, these will become much more enriched than your actual RNA because adapters are present in almost every sequence. So if you don't remove them, it is going to bias or skew your results. Third, because now tools have evolved to automatically detect if adapters are present, what it will end up doing is it'll put more workload on your computational power, and it'll unnecessarily increase the length of your sequence as well. And of course, it is not a desirable sequence, so we have to get rid of them. Apart from that, if you also have used any other PCR primer which has remained ligated to your sequence of interest, that also should be removed. Anything but your actual sequence of interest should get removed, should get trimmed, and anything which is actually a part of the human genome should be retained. This job is carried out by Trimomatic. Now we'll understand how and what are the different commands which actually make sure that this job is done correctly, and necessary sequences are not trimmed and unnecessary sequences are always accurately trimmed. So how does this program work? There are two major modes of this program. First, paired end, second, single end. Now, what do we mean by that? It actually depends upon what type of data you have. If you remember, I think I showed it in the last class where I was showing you how to retrieve data. There, if you remember, we had seen that many parameters of the sequencing reaction are often mentioned. What platform has been used for sequencing, what is the depth of sequencing, and most importantly, if it is single end or paired end data. I think in the previous class we had also discussed how uh, the two uh, the two mates of any sequencing read, how having both of those mates is important and necessary for multiple different purposes. We'll also understand why paired and data analysis is a lot more accurate, and it helps avoid getting rid of necessary sequences also. We'll look at it in a couple of slides. So the paired end mode will maintain the correspondence of read pairs and also use the additional information which is contained in the paired reads to better find adapter or PCR primer fragments introduced by the library preparation process. What do we mean by that? Having the paired end mode or using the paired end mode. When can you use it? When your data is actually paired end sequencing data. Under those circumstances, what will it do? It will help you better filter for any adapter sequences. You would see that some regions actually overlap between those paired and reads, and there are some regions which are present in only one and some which are present in only other, which are consistently present in multiple sequencing reads. Then that indicates an adapter sequence which needs to be chopped off, and the remaining valid sequence should be retained. Um, yeah, and these actually have gotten introduced during the library preparation step, which is something we have already discussed. Trimomatic works with FastQ files using FRED33 or FRED64 quality scores, depending upon the Illumina pipeline used. You're already familiar with what FRED quality scores are. And what does it do? Trimomatic preferably works with FastQ files, which have passed these quality scores, which is why using FastQC before Trimomatic is also important, so that you only use tools you only use those sequencing reads or those FastQ files which have passed this quality threshold, which means they're already of good quality. And now using Trimomatic to get rid of unnecessary sequences is going to further refine those reads for further analysis. Again, please interrupt me if I'm confusing you at any point of time. These are new topics, and even if you've used the tools, you might not actually have read all of the details. So I'm trying to explain that to you, but just let me know if I need to take a pause and repeat anything. So there are two major modes of the program, the paired end and single end mode. It does I think I just ended up copying this. Okay, so basically it works with FRED33 or FRED64 quality scores, depending upon what Illumina pipeline was followed. And I'll move forward to the next slide. What does it do? It uses all of these different parameters. Now keep in mind one thing. Whenever you are using any of these tools, uh, again, if you're comfortable with a Linux platform, it is going to be so much easier for you to perform analysis. If not, Mac and Windows are also usable, but it's more efficient using a Linux system. So if using Linux, you can use your command line and you can check for help. and it'll give you extensive detailed manuals for each of these tools. When you open up the manual, then you'll find all of these different parameters which will be given to you in detail. You will find what is the code you need to write for that particular parameter, and it will also advise you what is the most default parameter, what are the adjustments you may make depending upon what type of data you want further, what type of applications you have, etc. This is one major resource you should always use whenever you are utilizing any particular tool which you have not used extensively before. And even if you have, there might be a lot of things that you have not yet discovered. So if you just go through the manual, you'll find so much more to read about. Second resource, which you can often follow whenever you want to learn about the using a tool, if you're trying out different things and you're practicing analysis, then what you should look at are GitHub resources, because that is something you'll find open access most of the times. Secondly, you'll be able to, if you are somebody who has learned analysis and is a little bit more confident about it, then you can actually connect with the original authors of these manuscripts of these tools. And if you have any queries, if you want to modify anything based on your analysis, if you can collaborate, you can develop ideas, and it becomes so much more accessible. Additionally, on GitHub, you will also find detailed pipelines. You will find resources to study about these pipelines. You'll find resources which will tell you how that tool has been established, how much of it you can modify, how much of it you should not, because that is going to affect your result. And a lot of other information. GitHub it's an endless resource for bioinformaticians, and you can certainly refer to that. I highly recommend it. But for now, since we are following the textbook protocol, first of all, we'll understand what sort of parameters it can use, and then we'll understand the relevance of each of these parameters later on in more detail. So, the trimming step includes these different sort of commands. Let us understand them. The first one is Illumina clip. You'll understand how these are used. Just hold on for a couple more slides. So Illumina clip, as the name indicates, it basically means you are clipping off any Illumina derived adapter sequence or Illumina derived sequences. What do we mean by that? It means that within your FASTQ file, any sequence which gets incorporated into that sequence because the reaction has been performed on an Illumina machine will get removed. Basically, it is getting rid of any contamination which has come in from an Illumina source. Why only Illumina? Because Trimomatic is the most compatible with Illumina data. If you have used other platforms like we have discussed, if you've used Roche or if any other tool, then you can look for the most appropriate tool under those circumstances and accordingly modify the protocol. You can also use this Trimomatic itself also. You'll just have to play around with the parameters a little bit. But again, since we have been talking about Illumina from step 1, so that's what we are going to follow. The second parameter talks about sliding window. If you remember, we had seen it in this slide also. Yeah, here's where we saw sliding window. So what does it mean? It basically uses a trimming approach where it scans from the 5' end and keeps reading. It keeps looking like it sort of takes a window. And what does it do? It takes the quality of that read within the window. And if it passes a certain threshold, then that sequence is kept. If it doesn't pass that threshold, that's where clipping will be done. So basically, you are taking short windows. Think of it like how a ribosome moves over an mRNA transcript. What does it do? It takes a window of EPA sites of three ribonucleotides, three nucleotides. And then what does it do? Based on that window, it incorporates a particular amino acid, a charged tRNA carrying a certain amino acid. Very similar to that, using the sliding window approach, the tool basically scans through small regions of the entire FASTQ sequence, and it takes the quality score for each of those bases, and then it takes the average of those quality scores within that window. And if that quality score doesn't pass the threshold, then that's where it chops off the sequence, meaning that this is something of very low quality and this is something that is not actually a correct sequence and should be gotten rid of. Third is Max Info, which is an adaptive quality trimmer, which balances the read length and the error rate to maximize the value of each read. Then you have certain other parameters like the leading or trailing. Leading means you cut off bases from the start of the read. Like if you by default want to set a parameter that um, if you have uploaded, say, some sequences that you want to undergo trimming, that you want should undergo trimming, and you want all of the bases to be chopped off till their 15, if you want all the reads to be chopped off till their 15th um, base pair, then all the sequences will be clipped in this manner where the first 15 sequences or the first 15 bases will be removed. Same you can do from the end, and it will do so. First 15 bases will be removed only if there is a threshold which is not being crossed by that region, and that's where clipping will be done. You can also crop, where you can crop them to a specific length. For example, you can crop them to 80 base pairs, 90 base pairs, so on and so forth. Head crop means you want to cut off. Yeah, this is what I was mentioning. Actually, I mixed the two. Head crop is where you cut off specific number of bases from the start of the read, whereas where it means you can chop off the first 10 bases or 15 bases as per your requirement. Then you have min length, where you should drop a read if it is below a specified length. For example, if you only want to use 100 base pair reads, and if it is shorter than that, then the sequence will be discarded and not used for further analysis. Average quality is that if the overall average quality of the entire read is below a particular threshold, then that read will not be taken into consideration. Top fret 33 and top fret 64 converts scores to fret 33 and fret 64. That's just another parameter. So these are some trimming attributes which you can take into consideration. Let us now understand these in a little more detail. The first one is alumina clip. Again, something we have already discussed. What is its job? The job is to identify adapter or any other contaminant sequences within a particular data set. You want to get rid of anything which is not the actual sequence, and you want to trim them out, you want to trim them off. Now, this particular step is a trade-off between sensitivity and specificity. What do we mean by these terms with respect to this particular tool? Sensitivity means that the tool should be sensitive enough to find and to remove any contaminant sequence that is present. So its level or its accuracy of detection should be very high, where every time it comes across a contaminant sequence, it is able to detect it. Second, specificity means that it should not incorrectly get rid of actual sequence data, and it should leave all the non-contaminant and valid sequences within the data intact. That should remain untouched. This problem is even more acute when only a small part of the contaminant sequence is included within the read. What do we mean by that? Basically, what we mean is that when that sequencing reaction has been performed, if only a very short contaminant is present, sometimes it gets difficult to actually recognize that as a contaminant, and it might just seem like a variation from the human reference genome. And in those cases, because you would have low confidence over it being an unnecessary or a junk piece of sequence, you would not actually end up trimming it, and that makes it even more dangerous. You would end up retaining junk DNA within your sequencing analysis. The possibility of sequencing errors within the reads complicate the process even further. Although adapter and other technical sequences can potentially occur in any location within reads, by far the most common cause is the sequencing of a DNA fragment shorter than the read length. In this scenario, we'll come to it, don't worry. What do we mean by this? We mean that these adapter and technical sequences can occur anywhere within the read, but most commonly adapter contamination occurs because the DNA fragment that you are actually sequencing is shorter than the read length. So if your read length is supposed to be 100 base pairs and the DNA fragment that you have gotten after sonigation is 80 base pairs, for example, or 85 base pairs, for example, then what does the sequencing machine end up doing? The beginning of the read, it reads, it sequences valid data, but towards the end, it is not because it wants to satisfy that criteria of 100 base pair. The sequencing reaction keeps on proceeding and it continues to read through into the adapter. What does it mean? Apart from the valid sequence, the adapter sequence also gets decoded. This results in partial or full adapter sequence towards the 3' end of the read, and it is extremely common. While a full adapter sequence can be identified easily, identifying a short partial adapter is inherently difficult. So, interestingly, in a paired end data set, read-through will occur on both the forward and the reverse reads of a particular fragment in the same position. Also, because the fragment was entirely sequenced from both ends, the non-adapted portion will be reverse complements of each other. Now, since adapter read-through is a relatively common occurrence, and since Illumina datasets are often paired end, what does it do? Trimomatic includes another adapter identification strategy, which is called as the palindrome mode. We'll look at it also in the next slide. What does its job? Its job is to bypass adapter read-through and takes advantage of added evidence, which is available in paired end data. Basically, it looks at the presence of the overlap between these two sequences, and it is called as the palindrome mode. Have you understood till now? Please let me know, and then will I proceed, or I'll explain what I've already said in a better manner. Please let me know. Hello, am I audible?
Yes, ma'am. Yes, ma'am.
Okay. So, this shows you an example of how Trimomatic actually does all of this. So, we'll look at the legend and the actual text. So in panel A, what you can see is the read, which is shown here, and your aligned region, which is any technical sequence which you have taken into consideration. So if there is complete alignment of this particular region with the read, then you know that it is a completely detected sequence, and complete alignment has occurred, and this read can pass through. There is no contamination, nothing that needs to be trimmed under these circumstances. The second situation, where only partial alignment is seen for the technical sequence towards the 3' end of the read, and only therefore a short alignment can be seen. So look here, you can see that this is your technical sequence. Technical sequence basically means what you are using for, in this example, what you are using for checking the alignment and checking the efficiency of your tool. So if your technical sequence only aligns with a very short region towards the 3' end, what happens then? Below some length threshold, which depends on the relative cost of false positives and false negatives, it is not possible to identify an adapter sequence, and short adapters many a times will remain undetected. Like I just mentioned, if an adapter is quite long, then it is much more easier to detect, versus if it is a short adapter, you might end up not even recognizing it, and that actually can get retained, and that is actually possibly going to affect your data as well. So what does Trimomatic do to increase the efficiency of this trimming process? It goes for the palindrome approach. Now keep in mind the palindrome approach is applicable specifically only when you're using paired end reads, where you have two reads which overlap with each other. Paired end sequencing, where you have two reads which overlap with each other, and therefore you are much more confident in trimming of your data. So in this panel, the palindrome approach is used to check a similar situation with a short contaminant at the 3' end. What you saw here, if you have something similar here as well, where you have a small sequence, which is your junk DNA or your adapter DNA. Then, using this palindrome approach, what happens? You can see that because additional data is available in the paired end mode, the region tested as part of the alignment is much longer. So here you can see the region that is getting tested is aligning with a very small region, very small portion of the actual read, versus for paired end sequencing, because a lot more information is available, a longer region of alignment can be taken into consideration. So, yeah. Not only are both the adapter sequences tested at once, this is one adapter, this is another adapter, one for the forward read, one for the reverse read, but the fragment sequence also from both ends or from each of these reads are also getting checked. So this alignment, this trimming, this alignment, this entire analysis is a lot more reliable. A, because a longer region is being taken into consideration. B, because when you have such long reads for consideration, you are now sure that this exact green region is your valid sequence, and anything which is outside of that is not valid, is an adapter sequence, and should be gotten rid of. So, yeah, this alignment is a lot more reliable than the short alignment in B and allows adapter read through to be detected even when only one base of the adapter has gotten sequence. So that makes it a very sensitive and specific strategy for trimming. Why are we calling it sensitive? It is because the red region that you see here, the adapter sequence, if it is as small as one single base, even that can get detected for paired end sequencing. So that increases the analysis accuracy performed by Trimomatic. And also simultaneously, both adapters can get checked, which gives you more confidence in your data. And what you see in figure C shows how palindrome mode can also detect long read through. In this example, there's no usable fragment at all. Both of the reads begin with sequence from the adapters. This is still a sizable fragment, and this can be reliably identified. And you can see that even this is an adapter, and this is an adapter, both of which will get removed. And the correct detection of both short adapters and long adapter sequences, both of them can be performed very efficiently using Trimomatic. That is what we see here. Uh, Trimomatic uses a two-step approach to find matches between adapters and reads. What is this approach? How does this approach actually work? So first, short sections of each adapter, maximum 16 base pairs, are tested in each possible position within the read. If this short alignment, this seed alignment, is a perfect or a sufficiently close match, then the entire alignment between the read and the adapter gets scored. So basically, what do? You want to check for alignment between adapter and read. You will use a small seed, which is which you use for aligning between various positions. And if you see that there is alignment at any position, then that can get detected as an adapter start site, and that is going to help you understand that this is something that needs to be trimmed. Um, yeah, this two-step strategy. Yeah, the first step is basically you are using a short adapter sequence and you are testing every possible position. Second step is that if it is a perfect match, then the entire alignment will get scored. This two-step strategy is a considerable improvement for the efficiency of this particular analysis, since the seed alignment can be calculated very quickly, while full alignment score is calculated very rarely. Full alignment score, what do we mean by that? It is calculated as follows. Each matching base, every time there is, after complete alignment, every time there is a complete alignment between two bases, it is going to increase the alignment score by zero point 0.6, while each mismatch is going to reduce the alignment score by Q by 10. 1/10 of the quality score is going to get deducted. By considering the quality of the base call, mismatches caused by read errors have less impact. If there's a perfect match of a 12 base pair sequence, it will score just above 7, while 25 bases are needed to score 15. And the recommended range for full alignment between 7 to 15 is what you will usually find suggested as the threshold value for simple alignment mode. For palindromic matches, a longer alignment is possible. Therefore, this threshold can be even higher, somewhere around 30. Even though this threshold is very high, Trimomatic is still able to identify very, very short adaptive fragments, and that increases the efficiency of this particular technique. So overall, what do we see for Trimomatic? What are the properties that we saw? First one we saw is Illumina clip, whose job by default is to get rid of any known Illumina derived sequences, whether it is an adapter, whether it is a PCR primer, it is a sequencing reaction fallacy, or whatever reason it is, this tool is able to detect it and get rid of it. Second, what we saw is that Trimomatic uses this two-step approach. It basically first uses a small seed sequence, which tries to align at many different positions. And secondly, after there has been alignment or a sufficient alignment or a sufficient overlap between the scores, then it is able to detect the score. It is able to delineate a score for that alignment. And ideally, what is performed is alignment of that small seed instead of the entire sequence. If the entire sequence is aligned, then the score is calculated where every matching base increases the score by zero point 0.6 and every mismatch deducts it by 1/10 of the quality score. And overall, basically, that's what it does. It calculates how efficiently alignment can be performed. Second, this is how Illumina clip command works. Second command is sliding window. So what do we mean by it? Basically, it performs a sliding window trimming where it cuts once the average quality within the window falls below a threshold. Like I already mentioned, much like the ribosome, if it is taking a small window of 5 or 10 bases and it is calculating the quality score for all of those positions, and if it is seeing that the average for that particular position not able to um, is not able to pass a particular threshold, then that sequence is considered to be a contaminant or a junk DNA, and then that will get eliminated. And it'll get cut off and it'll get trimmed at that particular position. By considering multiple bases, a single poor quality base will not cause the removal of high quality data later in the end. So that is what the second command does. The third command is max info. What is it going to give you? It performs an adaptive quality trim where sort of balances the benefit of retaining longer reads against the cost of retaining any erroneous base calls. So you want as long a length of the read as possible. Why do you want that? You have um, See, if your reads are too short, if you simply tell Trimomatic that, trim all my reads where you remove the first 20 bases and the last 30 bases, and you only take the sequence in between, that is going to be a very easy job. But what is it going to do? A, it is going to give you very short reads. Um, Secondly, what it is also going to do is that the possibility of overlap between different reads is may or may not get reduced by a large extent. You eventually want some overlap between different reads so that you can stitch together these sequences finally with respect to the reference genome and align it. So if your individual reads become too small, then in that case, it is going to become very difficult to stitch them together. Secondly, they would be much more prone to, errors, to getting damaged, to getting chewed up, etc. And also, thirdly, you have paid a lot of money to get bases of a certain size so that your reads accuracy can be much higher and it cannot be, simply considered to be a, a sort of a fluke. So you don't want your sequence to become extremely small. Therefore, setting up the minimal read length is more often than not a good idea. The typically, the length which is required before a read is likely to be unique, depends upon the size and complexity of the target sequence, but usually the target length is somewhere in the order of 40 bases. The second parameter under max info is for additional read length. There may be added value in retaining certain additional bases because these are needed to place a read uniquely place a read. This is dependent primarily on the application. For counting applications such as RNA-seq, unique placement is sufficient. For assembly, variant finding, like when you want to check for mutations or variants, additional bases will provide more evidence for or against putative results, and they are more valuable. Third, you have error sensitivity, where you want your downstream analysis to be more or less sensitive to any errors within the data, which is determined by tools and the settings used. And Yeah, what does it do? It want you want tools to um one extreme would be tools where a single base error would cause the entire read to be ignored, which favors aggressive quality trimming. The other extreme would be tools which can tolerate even or even correct a large number of errors, which favors retaining as much data as possible. Just give me a second, I'm getting a okay. So yeah. So what was I discussing? The max info parameter of Trimomatic. So the first parameter was minimal read length, where you want your read to at least have a certain length, which can make it more reliable. Second, you can specifically ask the tool or set the command for the tool that you can have some additional basis depending upon what is your application. And third, you want the error sensitivity of your tool to be sufficient, whereby you want it either you would want it to perform very aggressive quality trimming, where you don't even want a single base error. And another extreme would be where you want to correct or you want to adjust with some number of errors so that more and more data can be retained. So these are different parameters. Now, somebody might, ask that why would you even want to do the second one? One of the reasons why you might want to do this is that if the data set that you are studying, if it is something which is very mutation prone, is very variant prone, then under those circumstances, getting rid of any data which differs from your original reference genome might actually end up compromising your experiment. You might lose out on a lot of data which you want to include because that is your actual objective. So optimizing the analysis based on your application, based on your hypothesis, is something very crucial. These parameters have been provided to you so that you can adjust. Now, somebody might ask, how would we actually know what is the correct extent which we should include, should not include? Majorly, what you would expect in these situations, either you should take the help of a bioinformatician, an expert whose job is to do these regularly, because they are capable of performing a lot of hidden trials where they can adjust different parameters. They can try the analysis of this three of these parameters and then give you the most optimal one. That is one way to go about it. The other way, of course, is to rely on available literature, available resources. Like I previously mentioned, GitHub is a very good place to get information like this because software developers, researchers, scientists, graduate students, just students, any student, much like you, who is interested in analysis, will be able to share their experience. And once you also are confident about any sort of data which you want to share, any sort of analysis which you want to, make publicly accessible and form more collaborations, you can do that as well. So these parameters which we are discussing are simply to understand how the tool works. How we should use it is up to us based on our analysis, based on our objective, and based on what sort of data we have. Another thing which I would also like to reiterate again, um, please keep in mind that for the same raw material, for example, if you are performing RNA sequencing of, say, for example, some bacterial cells which have been which are overexpressing something, and you are comparing it to the control condition. For this exact same experiment, if you get sequencing reactions done from multiple places, you might see a large difference in quality. Certain companies who are very who are well known, who are quite promising, and which also charge the moon, they are very good at it, and they will give you to some extent pre-processed data as well. I gave an example already of companies like Macrogen, companies like Arkinos, etc. They do this regularly, and you'll find very good data from these places. These are just examples. I'm not recommending any of them. I'm giving examples of companies who do. And there are some places which you might, get sequencing reactions at very cheap prices, but the quality of their sequencing is not very high. And so any data which you might actually end up concluding from those sequencing reactions might, under certain circumstances, be less reliable. So therefore, if you as the investigator, if you are analyzing both these types of data sets, then you should know that how much your pre-processing steps need to be relaxed or made more stringent based on what type of data is given to you. If your data quality is very high, then you can be more stringent. If your data quality is a little bit compromised, then it is more advisable to go for more relaxed alternatives, and preferably um, include more and more data. Now, the combined score is calculated for every possible read length, and the optimal score is used to determine which read or where within each read where it should be trimmed. Um, at very short read lengths, the minimal read length factor, it dominates. If you remember here, yeah, if you remember here, this was one parameter, the minimal read length. If your read length is very short, then the first parameter will predominate. What does it mean? It means that it will very heavily penalize those reads which are too short to be useful. Once the target read length has been achieved, the minimal read length factor penalty becomes a modest bonus. It's simply having an advantage. However, once the read is significantly longer than the target length, further bonuses from the minimal read length factor are limited due to logistic function. The additional read length factor then provides a benefit as additional bases can be retained. This is countered by increasing the penalty as the error free probability drops with increasing read length. I'll explain this, don't worry. The balance between these two factors is controlled by the strictness parameter. For most reads, depending on the quality of the read and the strictness setting, the increasing penalty from likelihood of error exceeds the bonus of retaining additional bases at some point, and the read is trimmed accordingly. So we said a lot of things. What does it mean? First, if you have very short read length, then having the minimal read length factor, this first factor, is very useful. Why? If a read is too short to be useful, then what will it do? It will heavily penalize that particular read. If you have a read where the target length has been achieved, then this factor is not very important. If it is very long than your ideal length, say for example, if your ideal length has been set to 100 base pairs, if your read is 180, 200 base pairs, 250 base pairs, then this first factor is not very important because the minimal length has already been achieved. In fact, it is too long. Then what do? You shift towards the next parameter, which is the additional read length factor, because additional bases have been retained, and then that becomes a benefit. But what do you do? With increasing read length, the possibility that that read is error free and most of it is actually correct, that will decrease. So in that case, this error sensitivity will go down because read length has gone further up. Basically, what you are doing is you are going to think of it as assigning a score. If a minimal read length has been met, then it is a positive. If an additional read length is too long, then it is a negative. Then error sensitivity is decreasing, that is also a negative. So basically, you balance all of these three factors so that your read length is optimal and the error sensitivity is also high, and it is the most as ideal as possible a situation for you to proceed with your analysis. The next parameter which is used when you are utilizing Trimomatic is using this term leading. We have already discussed this. You can remove low quality bases from the beginning, where you can set a threshold and you can get rid of any bases or any sets of bases within that window that do not meet that threshold, and then the next base will be investigated. Trailing means you are removing low quality bases from the end. If a base has a value below the threshold, then the base gets removed. The next base, um, which as Trimomatic starts from the 3' end, would be the base preceding the removed base, will be investigated. This is especially utilized when you are removing low quality segment regions, but most commonly sliding window or max info are used instead. Most commonly, what I have previously used also, and what you will find in most resources, is the use of sliding window. So yeah, apart from that, another parameter is using crop, where you can set the exact length to which you can crop your bases. So it removes bases regardless of what is the quality from the read end, so that the read has the specified length after cropping, and the steps which are performed after this crop might, of course, further shorten the read. Then head crop means you're removing specific number of bases irrespective of the quality from the beginning of the read. Then you have min-len, which removes reads that fall below that specified minimum length. If required, it would normally be after all other processing steps. Reads removed by this step will be counted and included in the dropped reads count. Again, something you will understand later on. What do we mean by this particular term? Normally be after all the processing steps. I'll show it to you in the next slide. This is an example of the sort of command that we give for Trimomatic. Do not get intimidated. Why? What you see, these terminology that you see in between this S11 sequence, S12, etc., blah, 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 till here. These are the names of your FASTQ files that you provide as input to the tool Trimomatic. Okay. For paired end Trimomatic analysis or Trimomatic based trimming, four sequences need to be provided, where you have here you can find S11 sequence text, S12 sequence text GZ, which is for the FASTQ file, and you have this forward paired, you have forward unpaired, you have reverse paired, and you have reverse unpaired. These are the four sequences that you provide to Trimomatic. Java minus jar Trimomatic 3 jar. This is the command line that needs to be followed. And here, if you have saved Trimomatic in any particular folder within your system, then you need to give the path for that folder as well. Then, after you have provided this information, you next move on to Illumina clip, which is the command which we have already given. Um, TrueSec is the library that has been used here. PE means it is a paired end sequence that has been taken into consideration. And this definition I'll come to when I explain here. Leading, trailing, sliding window, min-len. These four parameters have been mentioned. Now keep in mind, the order in which these parameters have been mentioned actually makes a difference. It dictates to the tool that when you are trimming each of these FASTQ files, you should keep in mind that this is the order I want you to follow. First, I want you to chop off the leading three bases, then the trailing three bases, then I want you to use sliding window tool, and I want you to investigate this window and then set up this threshold. We'll read the legend to understand this better. So when you give this command to the sequence, it will take to the tool, it will take these four input files and it will give you these output files. And what will it do? It will remove Illumina adapters provided in this particular file. Initially, Trimomatic will look for seed matches. If you remember, we had talked about seed matches of 16 bases, allowing a maximum of 2 mismatches. These seeds will be extended and clipped if in the case of paired end reads a score of 30 is reached, or in the case of single end, if in the case of single end reads, a score of 10 is reached. So this small text that you can see here, it gives all of this information. So what will it do? Illumina clip tool will look for Illumina clip command will look for TrueSec3 .fasta file based adapters, and it will get rid of any of the adapters which are present in this particular file. Where will you get this file? This is all available online. So whenever you want to get your sample sequenced, you will have information about what library is being used by the company that is performing the sequencing reaction. So sometimes you get TrueSec, sometimes you might have some NovaSec or some other library. So if you go to their website, you will get information about all of these tools and all of these libraries. You can download that, you can feed that as an input for the tool. And then what will it do? It will allow for a maximum of two mismatches, which is the first parameter you see here. Again, keep in mind this can all be adjusted based on what command you give um, to your tool, but these are the most default settings. Then the seeds will be extended and they will be clipped if it reaches a score of 30, or if it for single end reads if it reaches a score of 10. Then this is how Illumina clip is going to work. This is where Illumina clip command ends. If you notice carefully, till now there are no spaces, and that indicates that this is all part of the same command. After that, once you give a space, the next command begins. Then you can see leading, which means that it is going to remove any leading low quality or n bases below quality 3, which is leading 3. Remove trailing low quality or n bases, which is below quality 3 for trailing 3. If we had introduced a certain parameter like crop here, then that would have cropped the first x number of bases. For head crop, it would have cropped the first x number of bases. If we had used crop, it would have cropped all of the sequences to a certain length. So based on which commands you include and exclude, the tool is going to work accordingly. Um, yeah, then for sliding window, what is it going to do? It is going to scan the entire read with a 4 base wide sliding window, and it will cut where the average quality per base drops below 15. It is going to strike off, or it is going to trim the sequence right there. And finally, any read which is less than 36 bases long will be chopped off after all of these steps. Okay, so based on the order in which you write your command, the tool is going to follow it in exactly the same order, and it is going to carry out whatever instructions you have provided to it in exactly the same order. And usually minlen comes right towards the end. Before you actually use it, you want to use all of these other parameters, and then you want to get rid of any sequence which is beneath a certain length, so that it doesn't end up affecting remaining analysis. So this was an example of how you can or how you should give your command to Trimomatic. And I think, yeah, this will be my last slide for today. I'll explain this and then I'm going to show you the Trimomatic page for reference. So what we will begin in the next class is use of the understanding the reference genome in more detail. How was it structured? How it has been standardized? What sort of populations it includes, etc. And what is its relevance? Why is it such a big thing to have a reference genome, and why has it become so, like, in today's time it has become accessible, but how huge a project and how extensive a project it was. We'll learn about all of that, and what tools are actually used for aligning our sequencing reads to the reference genome, and how we use it further. All of that we'll start up discussions about that next class onwards. But basically, the idea of the reference genome, um, you want to identify, you want to describe changes in DNA, and for that, so that you can normalize that information for different people from different age groups, countries, populations, races, etc., etc., etc. You need to have one common standard sequence. What is that? That is called as the reference genome. Now, keep in mind, there are many, many variations to this. I'll discuss some of that. Many populations have standardized their own. For example, for a lot of cancers, a lot of diseases, you see some population specific biases. For example, African population has a different shows some disparity. The Indian population shows some disparity. Chinese does, etc. So there are differences also, but there is also a large portion of the genome that is very common and that is extremely similar among different populations. So for that, having a standardized representative sequence is extremely important. Where has it been derived from? It has been derived from the genome of multiple people, multiple individuals in a normal population, which acts as the baseline for comparing the genomes of different individuals. This artificially assembled sequence is known as the reference genome. The aim of the reference genome is to provide a consensus sequence to which patient data can be compared. However, it has significant limitations. The pooling of sequence information from only a small number of participants also ends up resulting in a biased representation of allelic diversity in the global population. Reference genomes provide a view of the architecture of the genome, comprising both genic and intergenic regions. These include repetitive regions, some of which are very challenging to assemble, like segmental duplications, centromeres, telomeres, satellites, mobile elements. In fact, till date, the complete reference genome or the complete human genome has not been sequenced. There are still some parts of it which remain a challenge, and some of it has gotten sequenced very recently and has contributed immensely to research and academia. Apart from that, population genomics, guided by reference genome, also aids the identification of classical genetic variants, which is what I was just saying, that any population specific disease disparity can also be identified using the reference genome, as well as structural variants, which are very difficult to detect in fragmented and incomplete references alone, but are very important for adaptation. So basically, any change from the generalized baseline of genomes can be studied using the reference genome as a comparison. So that was the idea that I wanted to discuss for today. I'll quickly show you this is the website which you can use for studying. This is Trimomatic. You can just write Trimomatic USA Dell Lab. This gives you the official information. You can read this paper as well, the original paper by Bolger et al., where they have first introduced a tool named, maybe I'll zoom in a little bit, yeah, a tool named Trimomatic for Illumina sequence data, NJ sequence data. Here's where you can download it. This is what I was talking about. GitHub being a very common and very important resource. So if you open GitHub, you will get all of this information about it. You'll find the protocol, the detailed protocol for this. You will see what is required. You will find details of codes, like already written codes you'll find here. Like, for example, what I just discussed here for yeah, and also what you can see here, if you have questions, don't hesitate to contact them. You can write to these authors. Most commonly for most tools, people are very, very interactive, very helpful, because as you can understand, this is not a technology that has been here for hundreds of years. Everybody is learning, everybody is trying to make the most of resources we already have. And so people are very eager to share. So you can always interact with authors, you can interact with collaborators from around the world. More often than not, they will respond. Um, but yes, do not bother them. You have to ask genuine questions after you have, done your best, and resources are unlimited. So like I already mentioned, if you see here, this is the input forward, input reverse, and what you get are output forward, output forward unpaired, output reverse paired, output reverse unpaired, all these sequences. This is your Illumina clip with the parameters that you will get, leading, trailing, min length, etc. Here you can see sliding window also, and what you get as the output. You can get that information here as well. For single end, you do things a little bit differently. You just have one input and one output file. And this you will find in more detail here. So for paired end, you will use quality trimming and adapter clipping as well. Um, this is what you see as the same parameters. It's the same thing, just written on their official website. And it shows you what all you can do using this particular command. This is for single end. These are the different parameters. A lot of this I've actually put in your slides as well. And for single end data, one input and one output file are specified. And for paired end data, two input files are specified, four output files, two of which are paired, two of which are unpaired. And you will proceed with the paired outputs for further analysis. And the unpaired output are reads that you should draw. So that's what you do. These are all examples. You can read up on them, which they'll give to you in more details. Again, just a quick reminder, you should always, preferably if you are using publicly available data sets for analysis, you should always preferably use paired end data. You've already understood to some extent why that is much better. And yeah, then in that case, you need to use this paired end mode. But if you are, if you need to use a particular data set and you want to proceed with it, you can certainly go for the single end mode as well. Here you'll find more information about these tools, some of which I have already mentioned. But yeah, pretty much this is what you can do. Also, another thing to keep in mind is that yeah, after you have completed trimming of your data, you need to again repeat the FastQC step to now check the quality of your data. Now, somebody might, you might ask, why do I need to do that? So most commonly after trimming, what you would want is, if you remember, mm, let me show this slide. If you remember our FastQC discussion from last class, then one of the parameters which FastQC gives as the output is also adapter content. I'll share my screen. Yeah. So here is adapter content. The last parameter after sequence duplication over represented sequence is an adapter content. So if there is a fallacy in your, or if you do not proceed with trimming, you will certainly see that this adapter content parameter is not being met. But after Trimomatic, you would expect all of these parameters to get checked, and after FastQC you should always validate that now your sequencing report is perfect or your quality report is perfect, and then you can proceed with analysis. So till now the workflow steps that we have done is hypothesis designing, actual sequencing reaction. We have discussed the sub steps of sequencing reaction as well. The output that you get from your company or whoever you're getting sequencing done from are your FASTQ files. You first check the quality of your FASTQ files. You might end up seeing, something like this on the left, a very um, a terrible report under most circumstances. Then you trim those reads and you get rid of adapter sequences, PCR primers, you get rid of any place which does not have a good quality, any place which is overrepresented, anything which is inaccurate, all of that you get rid of. And after you have done that, after you have done trimming, you again check the quality of your FASTQ files, and you should get a very prim and proper, good report of your trimmed reads. Then you use these trimmed reads for further analysis. You will align them, you will decode them, and then you'll do downstream analysis. So that is pretty much all I wanted to discuss for today. If you have any doubts, please let me know, and then we'll end our class for today. And we'll take up reference genome preparation, the Human Genome Project, and alignment of trimmed reads to the human reference genome in our next class. If you have any doubts, please let me know.
Ma'am, I have just one doubt. Like,
Yeah.
we are like discussing this as a analysis part of like the data which we have retrieved from sequencing. After that, we do all of these, na?
Right. So from the output, after you have gotten out your FASTQ files as the output of your sequencing reaction, then what
Mm.
do you do with the data? You have to perform these pre-processing steps.
Okay.
So under
Before,
those, we
like, it is
discuss two
it
tools.
it is before analysis. Like for analysis, we
It
have
is
to
a
follow these
part
steps.
of analysis. The entire
Okay.
process is a part of analysis, but first you have to get rid of incorrect and wrong data. Then you
Mm.
move on to alignment. Analysis, technically, if you talk about it, it starts after alignment has been done. So you have figured out where that read has come from. Now you make sense of what that read is actually telling you. That is the core analysis part. This is data pre-processing, alignment, then detailed analysis. But overall, if you think about it, if you have data in your hand, anything you're doing with that data technically is analysis only. But yeah, these are just loosely interchangeable terminology.
Okay, ma'am.
Okay. Okay, so I'll end the class for today. Thank you so much for joining. We'll meet again next week. Good night.
Thank you, ma'am.